COVID has been with us for almost 2 years now, and I have never seen an infectious disease that has been so politically polarizing. You can look from one news source to another and get conflicting information. So for this presentation, I have gone to the literature and I am using only peer-reviewed scientific studies that will be presented here. In addition, these studies have been collected in a Dropbox folder, and I will provide a link at the end of this presentation so that you can look for yourself and determine what the truth is. There's a few goals for this presentation. One is to understand the biology of the virus, how it attaches to human cells. This helps us understand the strategies to prevent infection. In addition, the technology behind the vaccines is also very similar to some of the newer technology to help improve Rett syndrome. So I'd like to spend a little bit more time on that because I think it will be important for our future to understand what's coming down the pipeline for therapies. We're going to cover a little basic science to begin with. As you remember from your science classes, there is DNA in the nucleus of the cell, and we're going to pretend the nucleus of the cell is above this black line I just drew. So in the nucleus we have double-stranded DNA that unfolds so that messenger RNA can be copied from it. The messenger RNA then moves from the nucleus to the cytoplasm, and there the ribosomes attach to the messenger RNA. They read the code so that individual amino acids are attached end for end to make up a long protein. So this is the transcription and then translation into a protein. Now, in Rett syndrome, we have a gene for NECP2, and it is not coding correctly. So what happens is we have a messenger RNA that isn't coding correctly, and we end up with a protein that is not the correct protein for the MECP2 to work. This same sort of circumstance happens when the virus infects the body and when we're making proteins through the vaccines. Now the difference is the virus and the COVID vaccines don't go into our cytoplasm, rather they don't go into our nucleus, but they stay in our cytoplasm. This is a very complicated slide, but we're actually going to go through it fairly quickly because we're going to spend more time on this portion of the life cycle a little later on. So the virus has a life cycle, and this briefly describes it. Most of you know this already. So we're going to start here with somebody who is generating droplets with a virus in it. Now this occurs anytime somebody sneezes, coughs, has loud voices, shouting. And then these droplets with the virus contained within them then land on somebody else in their eyes, their nose, or the mucous membranes of their mouth. When this happens, then the virus has a chance to enter our cells. When they do that, they essentially take over the replicating machinery of our cells so that they become little viral factories, and then they secrete hundreds of thousands of other viruses, which then can go through the cycle again with a sneeze and infect another person, and this happens over and over again until we break the cycle. This is a depiction of the SARS-CoV-2 virus. This is the virus that causes COVID. And in this picture, you can see we have a viral capsid, which is a lipid membrane, and in this lipid membrane are all of these proteins that stick out that we call spike proteins. And inside this lipid capsid is tightly woven RNA. Over here is a close-up of one section of it. And the spike protein is what allows the virus to attach to our cells and get into them to replicate. The portion of the life cycle that you saw earlier, and I'd like to spend just a little more time so that you understand how the virus attaches to cells. So here is our COVID virus up here, and on the outside are all of these spike proteins like we saw in the previous slide. These spike proteins have developed by evolution to be able to attach to a receptor on our cells called the angiotensin converting enzyme type 2 receptor, or the ACE2 receptor. Once these spike proteins attach to this receptor, then they are able to be pulled through the membrane of the cell into their cell cytoplasm. When this happens, the lipid coating on the virus then opens up, and the messenger RNA that is inside of that lipid coating is able to start replicating in the cellular machinery of our own cells. So instead of our cells doing what they normally do naturally, they stop, and they are now viral factories. So there are several different proteins that are copied, and with all of this instruction, the cell can then make more proteins, make more RNA, make new lipid coatings, including the spike protein on the outside, and then release these into our mucous membranes. So this is a very efficient process once it starts, and this is why we have difficulty with these viruses. Now, the viruses continue to have little mistakes. That's most of the time those mistakes don't help the virus replicate, but occasionally, 1 in billions of the mistakes will cause the virus to be more effective in either attaching or making more viruses or something else that helps the virus compete. These we call the variants, and of course the latest one that we're worried about is the Delta variant, which is very good at attaching to the ACE2 receptor and very good at making many more copies of itself in the cell. The ACE2 receptor, or the angiotensin converting enzyme receptor type 2, is where the virus, of course, loves to attach to in our bodies. We have ACE receptors all throughout our bodies, but they are in higher concentration in our lungs, in our heart, in our vasculature, also our GI tract. Because of this, this seems to be where we have most of the difficulty when we contract the COVID virus. So, of course, we all know there's terrible respiratory failure from our lungs. We have trouble with pericarditis and myocarditis. And also there's thrombosis in the blood vessels, in addition to significant kidney issues. So that explains somewhat why we have these particular areas of concern in the COVID virus. That has gone into understanding the virus, its life cycle, is helping us really figure out how to prevent COVID infection. So the strategies that are being used that we hear about all throughout the news include masking and avoidance, a lot of therapies directed at blocking the spike protein, other therapies at blocking the target of the spike protein, the ACE2 receptor, and also looking at interfering with the replication machinery once the virus enters the body. Know that this is my only mask slide in the whole presentation. But before we talk about masks, Let's first talk about crowds and avoidance. We know that with loud noises, shouting, droplets that carry the virus can travel about 6 feet. So decreasing the number of large crowds we're in can be very helpful. Now, when we talk about masks, there's been a lot of controversy in the news about masks. Let's think about the two reasons for masks. One is to block droplets from leaving one person's mouth and going into someone else's eyes. The other purpose for masks is to filter the droplets so that they don't get to your mouth and eyes. So that's where some of the controversy comes from. When we're talking about preventing droplets, certainly even cloth masks will be helpful for that. When we're talking about preventing droplets from getting into our mucous membranes, the tighter the mask, the better. In general, studies have been done looking at the effectiveness of masks. And when we talk about preventing droplets from getting into our mucous membranes, there's a hierarchy of what works well. Certainly the N95 masks work best. Then we go to procedure masks that are the ones with the little loops behind the ear most of the time. Cloth mask, of course, there's all kinds of different cloth masks, but certainly a couple of layer cloth mask is pretty helpful at preventing transmission, so it is worthwhile. Gaiters are somewhat less beneficial, and almost of no benefit is a face mask without any other covering for preventing getting droplets in your mucous membranes. So they are somewhat effective. They're not 100% effective, but especially in big crowds, I do. At the start of this pandemic, we were very lucky in that there was a lot of research on the COVID virus family already going on because of the scares for SARS and MERS. Because of this, people understood and were able to look at the different areas the virus used for replication. So early on, there was a scramble to look at what off-the-shelf existing medications may help us prevent infection. This is a very busy slide, but it shows with the virus life cycle where people were considering using off-the-shelf medications once again at the start of the pandemic. So there are a list of medicines that may prevent the binding of the spike protein to the cell. And as you can see, chloroquine, hydroxychloroquine were two that were being considered. And then with all of the antiviral therapies that have been used throughout the years, they had some possible antiviral medications in this area, all part of what I call the cellular machinery of replicating the virus to consider. Now, many of these medications were tested on cells outside of the body with encouraging results, and they went on to peer-reviewed, double-blind, placebo-controlled trials at the start of the pandemic, with a lot of hope that they would pan out as beneficial. Same slide as the last, with the exception or the addition of those medications that did not prove effective lined out. So from all of these initial ones that were encouraging, remdesivir is the one IV antiviral that is indeed in use and helping to decrease the severity of COVID infection. In the last couple of days, the European Union has approved molnupiravir, which is also an antiviral that inhibits the machinery of replication for the virus. Now, the other exciting thing about this is I'm sure you've heard on the news, is it's an oral medication as opposed to the IV medication. So it can be used earlier in the course, and it's hopefully going to keep people out of the hospital. So this is an exciting addition. I also want to talk about ivermectin for a brief second. Ivermectin was initially found to be beneficial. This was based on a type of study called a meta-analysis. Now, a meta-analysis looks at several smaller studies and combines them in a statistical format that lets you predict with more accuracy whether something is effective or not. Unfortunately, the first meta-analysis that came out that showed that ivermectin was effective was flawed data. It turned out the biggest study of all of the studies in this meta-analysis was fraudulent. The researchers plagiarized data or information, and their data was completely made up. In fact, some of the patients they reported on had actually died of COVID before they even started their study. So once this data was retracted and the meta-analysis was reviewed again, it turned out that there was no benefit from using ivermectin. The most effective strategies to prevent COVID is to block the spike protein. We have several ways we can do this, of course. In the laboratory, we can make specific antibodies to the spike protein, give those intravenously to patients. That way they'll attach the spike protein, and then the spike protein won't be able to attach to the ACE2 receptor and go into the cell for replication. Another strategy that's used is convalescent serum. We can take blood from people who have recovered from COVID, isolate their antibodies, and give those intravenously to other patients who are sick and at risk of dying with COVID. This is beneficial, but not perfectly effective. For most of us, we need to focus on making our own antibodies to spike proteins. We can do this either by receiving a vaccine that will develop antibodies to the spike protein, or we can get COVID and have natural immunity, both of which will make antibodies. In my opinion, it's a lot nicer to get the vaccine because we don't have as great a risk of side effects. Antibodies are one of the tools we have in the tool chest. They're developed in the laboratory. The idea of the monoclonal antibody is they will attach to the spike protein. Here's a close-up of that. And when they do that, the spike protein of the COVID virus will not be able to attach to the ACE2 receptor, and so therefore it can enter the cell and replicate. So that's one of the strategies we use when we have very sick people, those that haven't been immunized and those that are immunocompromised, to keep their very serious illness from progressing to requiring intubation or death. They are effective, but they're not 100% perfect. Currently, we have 4 different ones that are approved, and they, of course, are very beneficial, but occasionally are in short supply as the number of people with COVID increases. I'm on this slide. Although throughout the world there are at least 20 to 25 different vaccines that are either being investigated or approved, in the United States we have 3, and they use 2 slightly different strategies. So we're going to talk about those 3 together. So the Johnson & Johnson virus uses a viral vector where they use actually a different virus than the COVID virus, an adenovirus type 26, which they then place the DNA for the spike protein into the virus's DNA. Now, this is very similar to a lot of the therapies that are being developed for Rett syndrome, where they're using an adeno-associated virus, which is related quite closely to this virus, to enter the cell and put either DNA or RNA into the cell. So with the Janssen immunization, what happens is you get this adenovirus that's been what they call attenuated, so it can't cause infection. It injects the DNA into the cell. The DNA then moves into the nucleus of the cell. It doesn't incorporate with the DNA of the person, but it sits in the nucleus. And from that deoxyribonucleic acid, as we saw in the very first slide, ribonucleic acid, in this case we call it messenger RNA, which is the RNA that codes for proteins, then moves out into the cytoplasm where the messenger RNA is turned into the protein for the spike. The spike protein then goes to the surface of the cell. Our bodies are very good at recognizing something that's foreign and making antibodies to it. So then the spike protein is recognized as foreign, and the immune system, in a very complicated method, makes... This is a very busy slide, and I apologize for that. In this slide, we're looking at serious side effects of both the COVID vaccines and getting the virus. We're not talking at all about the milder, short-lived side effects of fever, sore arm, myalgias, things like that. And going through this list, in the center here are the frequency of these side effects by different type of vaccine. So this is the Pfizer, this is the Moderna vaccine, this is the Johnson Johnson. And so there's a P, an M, or a J behind every one of these. In this list is the frequency per million of acquiring one of these problems when you have COVID. It's very important to know that this data is harder to come up with because there aren't a lot of great studies looking at the number of people who have gotten COVID, but there's a lot of studies looking at people who have acquired these side effects from COVID. But in every case, with the exception of anaphylaxis from the vaccine, you are much more likely to have these serious side effects when you have the virus than when you have the vaccine. Certainly we're concerned about this, but it's definitely safer to be vaccinated than to acquire COVID. Fortunately, most people that get COVID are not going to have these serious side effects. Finishing this presentation, the Pfizer vaccine for ages 5 to 11 was approved. I think this is an exciting step forward, and I strongly recommend our children get this vaccine. Having said that, it's important to point out that the data for approval was based on 2,268 participants. So this is a relatively small group, and the extremely rare side effects that we see with the vaccine in the ages 12 and up have not been seen yet with this vaccine, simply because of the numbers. I fully expect we will see very similar side effects to what was presented on the last slide, and I would expect they will be in about the same percentages. This is based on the fact that the immune system for 5 to 11-year-olds is very similar to the immune system of 12 to 20-year-olds. I want to talk briefly about multisystem inflammatory syndrome in children, MISC. This is basically an overreaction of our immune systems in response to the virus. When this When a virus infects a cell, the cells send out all kinds of chemical signals called cytokines to trigger other cell infection-fighting cells to come into the area and destroy the damaged cells. When there's an overreaction of the immune system and there are way too many infection-fighting cells coming in to fight the virus, this is called a cytokine storm, and sometimes the immune system gets out of balance and attacks a lot of our own organs, such as the lungs and the heart and the kidneys, and can result in death. This happens in children, fortunately very rarely, but happens with COVID. We have not seen it with the vaccination. This also happens in adults. So a lot of the therapy to prevent death in COVID is not to prevent the COVID infection, but to suppress the cytokine storm afterwards. Again, my apologies for a very busy slide. We're going to go over this briefly. So over here we have the various cytokines that we talked about that are overreactive in cytokine storm. And here's a list of medications that suppress each one of these cytokines. As you can see, there's an MAB at the end of most of these. That indicates that they're monoclonal antibodies that have been developed in the laboratory to attach to each one of these various cytokines in the pathway. There's also a lot of strategies for inhibiting the signaling pathway within the cell that are in this level. And down here are what I call the sledgehammers of the immune system. These are medications we've used for a long time, and they have very broad suppression of the immune system. So we've all used steroids in asthma and other situations. We've often used intravenous immunoglobulin to suppress the immune system. Also, there's some interesting work on some traditional Chinese medications that do that. So overall, we have several different strategies for suppressing the cytokine storm. The trick is to recognize it early enough. Of note, COVID is not the only virus that causes cytokine storm, and everything we are learning today is going to help for the next virus that comes along. And this is improving every month. There are new therapies for suppressing this cytokine storm. We've already talked about convalescent serum. Once again, this is just another way to prevent serious COVID disease. Well, this is the last slide in the pack. Thank you for listening. I wanted to spend a brief moment on supplements for COVID. There's been a lot of talk in the news about this, but when you look at the literature, there is no clear evidence that higher than normal doses of supplements are helpful in preventing COVID. So I do recommend zinc, vitamin D, and vitamin C on the daily recommended doses, but I don't recommend higher amounts. There are undoubtedly many more concerns about the COVID virus and vaccine. I could not take the time to list them all here. In general, as I've looked through the data, I do support getting vaccinated as a much safer approach to fighting this virus than getting the actual infection. Once again, thank you for your time, and I am always happy to answer individual questions.